So we pick up after the nucleus with our cytoplasm. Cytoplasm, like the cell membrane, is going to be another structure that we find in every single type of cell. Cytoplasm is the jelly-like fluid that fills the cell. It's going to hold your organelles. A lot of chemical reactions are going to occur here. It's basically the medium or the substance through which reactions occur, your organelles exist, transport occurs. It is a much better medium than an air would be. So we find that it is a water-based. So it's mostly made up of water and, again, found in every type of cell. The next type of structure, so again, this is not an organelle, it is a cell structure, is a ribosome. Ribosomes are also found in every type of cell. And the reason that we find ribosomes in plants, in animal, and bacterial cells is because they do such an important job. And they are actually the place where the proteins get put together. So back in DNA, we talked about how your DNA is important because it contains little sections in it called genes. And those genes are the recipes for how to make proteins. We said proteins, super important because they're going to make your enzymes. They're going to physically structure your cells. They're going to do all of these very important things. Oh, that was a bell. But when we look at where these are actually put together, we find that it's at the ribosomes. And there's two different places inside the cell where we can find ribosomes. So in all cells, prokaryotic and eukaryotic, so bacterial, plant, and animal, ribosomes are free in the cytoplasm. So oftentimes they're drawn in diagrams as tiny dots. So if you look over on the diagram I've given you guys, they're referred to as free ribosomes. They're just floating around in the cytoplasm. However, in eukaryotic cells, so now we're talking only plant and animal cells, we can also find ribosomes bound to the ER, or the endoplasmic reticulum. And what the ER is, it's essentially just a conveyor belt that's going to pass these little segments, these little recipes from the DNA that have been turned into RNA, because DNA can't leave the nucleus. So these are going to pass by these ribosomes, and they're going to get made into proteins inside the ER. And the ER is going to transport them a little bit later on to the Golgi apparatus, and the Golgi is going to package and ship these finished proteins wherever they're going, whether it's inside the cell or outside the cell. The next structure we're going to look at is the lysosome. And what we're going to do for the lysosome is we'll actually watch a short video, and I want you to consider what's the function of lysosomes and what do lysosomes contain.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
So if we look at what lysosomes do, so what is the function of lysosomes? Lysosomes are going to be filled, I'm answering the second part. I'll do them at the same time. So lysosomes are filled with enzymes. And the reason they're filled with enzymes is what they're going to do is just break stuff down. They are the janitors of your cell. So if it is a virus or a bacteria, so something bad that gets in, the lysosome can go in, take the enzymes, and disassemble that whole structure so it's not dangerous anymore. But it also breaks down garbage in the cell. So if a mitochondria is really old, and this was the example from the video, and it's not working properly anymore, the lysosome will go and fuse and use its enzymes to dismantle that mitochondria. The next one we have are vacuoles. So vacuoles are the closets of the cell. They're going to store cellular materials such as water, salts, proteins. We find that a big difference we have between them is that animal cells are going to have many small vacuoles, and your plant cells are going to have one large central vacuole that they fill with water. There's a hint over here. Just when you're looking at diagrams, lysosomes have enzymes and are drawn with materials inside. So a lot of times if you're looking at a cell diagram, your vacuole and your lysosome, especially with animal cells, are drawn as just small circles. That's how they're drawn. But if they draw something on the inside, they're typically trying to show, oh, these are enzymes that are filling this. So that's a lysosome. But we typically, when the vacuoles are drawn, they're drawn as being empty. To show this is the empty storage space of the cell. An interesting thing about the central vacuole in plants is that plants are going to overfill this central vacuole with water. And what's going to happen is that's going to actually cause the amount of water inside the central vacuole to push out on the cell wall. So actually put pressure on those thick cell wall structures. And we call that pressure being sort of forced on the cell wall by that overfilling of water in the center, turgor pressure. And turgor pressure is actually partially what gives our plants their structure. So we all know, I've given you an image over here on the left-hand side. We see a plant that's been nice and watered, so it's standing up straight. And then next to it, we see a plant that has not been watered, so it's wilting. Wilting is the result of a decrease in turgor pressure. So the central vacuole loses water. It doesn't push out on the cell wall as much, so you don't have as rigid of a structure to hold up that plant. So it wilts. It kind of falls over a little. You water it, and we know that those central vacuoles are going to fill back up. Turgor pressure will push back out on the cell wall, and our plant will stand up straight again. The next structure we're going to look at is the mighty mitochondria. So mitochondria produce energy in the form of ATP for the cell. Typically, students want to tell me that mitochondria is the powerhouse of the cell. You have to be able to explain what you mean when you say that to me. So if you just tell me it's the powerhouse of the cell and you kind of can't back up what that means, it's no good. You need to be able to tell me the reason it's the powerhouse is because there's this molecule ATP that's super important in cells because it's actually going to be the currency that runs all of the chemical reactions, runs all the processes in the cell. The mitochondria is the place where this molecule is made. So it's producing all of the power for the cell to be able to run itself. This is such an important job that we find mitochondria in both plant and animal cells. The next thing we're going to look at is a chloroplast. So chloroplasts are going to capture sunlight. So they capture energy from the sun, and they convert it into chemical energy using photosynthesis. So this is such a huge job that's being done here by the chloroplast. The chloroplast is taking sunlight, which is unusable energy for the vast majority of life on this planet, and it's converting it and storing it in a form that is usable to other organisms on this planet. And that form that it's being stored in is the carbohydrate glucose. So that's what is made in the chloroplast. So that energy from the sun, when it gets captured, that energy is stored in the bonds in between all your carbons, hydrogens, and oxygens that in that molecule of glucose that's going to get made. That glucose can then, it's a little stored piece of energy. So it can either be broken down by the cell later on, the plant cell later on, in the mitochondria to make energy for that cell, or a heterotroph, so an organism like ourselves, can eat this plant or eat another organism and basically borrow their glucose for us to break down in our mitochondria for energy. We know that chloroplasts are only found in autotrophs, so self-feeding organisms that can make their own food, so they are not found in animal cells. So I want you to think about it for a second. So it says prokaryotic cells lack mitochondria and chloroplasts. Does this mean bacteria cannot make ATP or store energy from the sun as sugar? So hopefully after thinking about it, you were able to get that no, we know that bacteria can be heterotrophic or autotrophic. So what it means when we say that bacteria can be heterotrophic and autotrophic, even though they don't have mitochondria and chloroplasts, it just means they don't have a specialized structure to perform these jobs. Bacteria are still going to make ATP. Some of them can still be photosynthetic, so they can still capture energy from the sun. They just don't have special structures to do it. They just do all of that in the cytoplasm. So the analogy that's often made here is bacterial or prokaryotic cells are like a studio apartment. And your eukaryotic cells, like your plant and animal cells, would be like a mansion. So here's the analogy. In a studio apartment, you have one big room that you do everything in. So you cook in your kitchen in this room, you're going to sleep in your bed in this room, you're going to watch TV in this room. Whereas in a mansion, you have different rooms. So you have a TV room, you would have a separate kitchen for cooking, a separate bedroom for sleeping. This is like prokaryotic cells do everything in their cytoplasm because it's the only room they have. Eukaryotic cells are like the mansion where they have special structures or special rooms to do special jobs.